Today's video is very much important for all the science aspirants. Those who are right now in class 10th, either in class 12th, or either they are preparing for NET or NEET examination.
Monohybrid cross.
Discussing about the cross. Now I will take the example of the Mendel's cross because that is something that always occur in each and every class like 10th, 12th or even for NET and NET preparations. So if we consider that Mendel's cross, then first of all we need to take the parental generation, because for every cross we first go for the parents, right? So the parental generation is the generation where we will consider the pure forms. What do you mean by pure forms? When there are homozygous conditions. And about homozygous and heterozygous I have already discussed in one of my video. So I'll share the link of that video over here. Now if we talk about the homozygous pure, then let's say this capital T means homozygous tall father is there and homozygous dwarf mother is there, and I'm crossing both of them, the mother and the father. Now they will first form the gamete. In the formation of the gamete there will be one capital T that will be crossed by, in this the gamete will be formed as small t and small t. Now after forming these gametes, we will form a Punnett square. Now let's understand how to form a Punnett square for a monohybrid cross and what are its details. If we talk about a Punnett square, then in Punnett square we actually form 1, 2 and 3 rows and 1, 2 and 3 columns for monohybrid. Always focus on monohybrid. And here we actually consider that this is going to have the male gametes and this portion is going to have the female gametes. This will have the male gametes and this will have the female gametes. Now this was my mother as we have considered initially, and this was the father, right? So now in the male gamete I will write these gametes, the gametes formed by the father, and that will be capital T and capital T. Whereas in this one I will keep the gametes formed by the mother, that is in female gametes small t and small t. Now when you will join these gametes, the fusion of gametes will occur for the formation of the progeny. So the fusion is going to occur, so one capital T will come from here and small t will come from here, and this is one of the probability of the genotype in the offspring. The another is, again we will take capital T from here, small t from here. Again we will consider capital T from here, small t from here. Again capital T and small t. So now the genotypes that we have got is genotype and phenotype. You can watch my video on genotype and phenotype, and from there you can understand these two terms. So if you will talk about the genotype over here, then genotypes are capital T and small t in all the progenies. So if we will talk about the genotypic ratio and the phenotypic ratio for this generation is the F1 generation, filial 1 generation. So for the filial 1 generation, the phenotypic ratio and genotypic ratio, if we will consider the genotypic ratio, then because all are having same genotype, so it is going to be 4:0, and that 4 genotype is capital T and small t, that is heterozygous condition for the tallness. Whereas the phenotypic ratio is also going to be 4:0 because the phenotype, the expression is all are going to be tall. So that was all about the monohybrid cross from parental generation to the F1 generation. Till now we have talked about the parental generation, the F1 generation. Now we will talk about the F2 generation, which is going to be obtained by crossing the F1 generation progenies. So my F1 generation progenies all were having this kind of genotype as we have discussed just now, and they all are going to be tall. So now for F2 generation, I am going to do a selfing. Selfing means I am taking the same progenies and I am crossing those progenies with each other, right? So I will take, because these are plants, so we can do that. So these plants, in this pea plant, we will take one of the progeny and do its selfing with another progeny. Now let's say this is going to be the male and this one is the female one. Fine. So again, now this is the F1 generation progenies and I am crossing these progenies. So again there will be the gamete formation. When the gametes will form, there will be two kinds of gamete that we will obtain, capital T, small t. Again here capital T and small t. Now we will form a Punnett square of this. So again, if you can recall, we have to have 3 rows and 3 columns, and it is going to have male and here it is going to have the female gamete. So here, considering this one as male and this one as female, the gametes are going to be same. So capital T small t and capital T small t. Now when you will fuse these gametes, then you will find capital T from here and capital T from this side, capital T from this side and small t from this side, capital T from this side, small t from this side, and small t from both the sides. So now in this F2 generation, you can see that there is a different scenario, right? Considering this scenario, if now we will try to calculate the genotypic ratio and the phenotypic ratio for this F2 generation, that is filial 2 generation, then what you will find that there are different kinds of genotypes, like one genotype is capital T, another genotype is capital T small t, which is common in these two, and the third genotype is of this kind. Right? So we are having 3 kinds of genotype. So in genotypic ratio, this kind of genotype is in 1 copy only. So we will write 1. This type of genotype is in 2 copies. So we will write is to 2. And this one is again in 1 copy. So is to 1. So 1 is to 2 is to 1. That is going to be the genotypic ratio, where this one is capital T, the homozygous dominant. This 2 is capital T, small t, the heterozygous tall, and this one is small t, small t. Whereas, if we consider the phenotypic ratio, then this is going to be tall, this is also going to be tall because T is dominant. This is also going to be tall, but this one, as it is recessive, homozygous recessive, so it will be dwarf. So, because 3 are tall, so we will write 3 tall and 1 dwarf among these 4. So, 3 tall and 1 dwarf, and that is going to be the ratio, phenotypic ratio. That is 3:1. So, in monohybrid cross, in the F1 generation, all phenotypes are going to have capital T, small t as genotype and tallness as their phenotype. Whereas in the F2 generation, the scenario is different, and that is, it is going to have 1:2:1 as the genotype and genotypic ratio and 3:1 as the phenotypic ratio. That's all about the monohybrid cross. Next video, I'll discuss about the dihybrid cross. In case of any question, you can write a comment and like, share and subscribe my channel. Thanks for watching.